So hello, all the listeners out there. I'd like to welcome you to our Hordes Dairyman in University of Illinois webinar. We have a good audience group out there listening, and we're really excited to share some good information with you today. My name is Abby Bauer, and I'm an associate editor for Hordes Dairyman magazine. I'd like to thank you for joining us and being a part of this webinar. I am fortunate to have Mike Hutchins from the University of Illinois with me as a co-host. And together, we have the pleasure of welcoming Dr. Marcus Curley, who is the director of the USDA's National Animal Disease Center. He will be presenting today for us, and his presentation title is Bolstering Transition Cow Immunity. The sponsor for this month's webinar is Diamond V. You see their logo there on the screen. We're very pleased to have their support for this month's program. I would also like to give a shout out to our other team members, Jim Baltz at the University of Illinois, and our Hordes Dairyman online media manager, Patty Hurchin, who are working behind the scenes to make sure everything runs smoothly for today's presentation. If you're listening live, you have access to a PDF of the slides that can be printed out. If you look at the GoToWebinar panel and scroll down near the bottom, there's a section that says handouts. And if you click on that, you have the option to click on some links and you'll see the presentation slides there. And that's if you want to be able to print out the slides for future reference or to take notes today. Mike, I think that's everything I have for now. So if you would please further introduce our speaker and let's get this webinar started.
Well, very good, Abby. It is my professional honor to welcome our distinguished instructor here and webinar leader, Dr. Marcus Curley, Jr. He was born in Massachusetts. We won't hold that against him. He grew up in Iowa and received all his degrees at the Iowa State University, getting his doctorate of veterinary medicine, PhD degrees there as well. Abby already has indicated his title as the director of the disease center there and has worked 36 years on infectious and emerging diseases as far as that goes. And some of you had a treat to hear a little bit about some of the things he discovered and talked about when he was over in China as far as that goes. He has demonstrated the importance of immune function in transition cows and susceptibility to such diseases as mastitis and metritis retained placenta. He's got a whole list of accolades here, but he is internationally recognized. In fact, he just returned from China on Saturday night here, so we're pleased he got back here. And certainly, Dr. Curley, we are so pleased to have you on. We'll turn the program over to you on the title that Abby already announced. Take her away, Dr. Curley.
Okay, thank you very much, Mike. As Mike indicated, I just been in the United States now for about a grand total of 36 hours, flying back from China, and my arms are tired. So with that old Johnny Carson joke, I will move right into our presentation today. So dairy cattle are a very profitable part of the United States and any country's ag economy. And the transition cow, as anybody who has ever worked on a dairy farm, having grown up on myself, you know that those animals are very fragile, that if they can get through the transition period without suffering disease, that cow is generally going to be a very profitable animal on your farm for that lactation. If, however, she experiences postpartum diseases, then it's going to be an economic burden for you if she survives. So much of what I'm going to talk about today is a large body of research that has been conducted and published from around the world. And we were front center involved in much of this. So I want to just jump right into that. And today I'm going to focus on these three questions. What disease challenges do transition cows face? Why do these diseases happen? And maybe more importantly, what can we do about it? So I'm presenting a slide here that Dr. Larry Smith at The Ohio State University published back in the mid-1980s. And I find this a very important piece of information when it comes to understanding some of the epidemiology of mastitis in a dairy herd. This was a large study that he conducted over a period of years. And on the y-axis here, it talks about intramammary infections. That's just the presence of bacteria in a quarter. And he has demonstrated that in these bars, this bar graph, that the first 2 weeks after calving and the first 2 weeks, the first 2 weeks after dry off and the first 2 weeks just before calving are when cows experience the greatest rates of new infections in the mammary gland. So obviously, those are a concern. And in fact, if a cow maintains an infection at the point of calving, a very high percentage of those cows will actually develop clinical mastitis in the first month after calving. These new infection rates are higher than any other time point during the dry period, as well as during lactation, being at least 2 to 6 times greater. So why is it these cows are getting these new infections? That's an important question. And if you look at when clinical mastitis occurs, data from various countries around the world are all been very similar. We know that up to a fourth of all clinical cases of mastitis, and in this slide here, we're talking coliform mastitis, those occur in the first week or two of lactation, nearly half in the first month, and almost 2/3 in the first 60 days after calving. So you get a lot of new infections occurring right after dry off, another high period of new infections just before calving, and then after the cow calves, you get a very high rate of new clinical cases. This is true on well-managed dairies. Data also out of the Ohio State University looked at commercially operated dairies in their area, and these were very well-managed dairy herds. Over 80% of the cows had linear scores below 5, but they still had unacceptable frequencies of intramammary infections caused by mastitis pathogens that would lead to clinical mastitis. And again, 20% of these cases occurred, of clinical cases of coliforms and environmental streps would occur during the first 7 days after calving, totally consistent with the research data that have been previously published. So this begs the question, are transition cows immune suppressed? Most pathogens that cause mastitis in a dairy cow can be considered opportunistic pathogens, meaning they don't really have a whole lot of ability to cause disease if the immune system is working well. So we have evidence that there's a high rate of new subclinical infections prior to calving, followed by a high incidence of clinical mastitis afterwards. The question becomes, does immune suppression convert these subclinical infections into clinical disease? And I'm going to go through some evidence that supports that this is indeed the case for mastitis, retained placenta, and metritis. I would also suggest that there's a high incidence of Johne's disease becoming clinical, as well as salmonellosis in early lactation. And these may all well be related to immune suppression at calving time. So we're going to have a little bit of Immunology 101 today, and I think it's important to understand just some very simple concepts here, but there are two major branches of the immune system. One we call the acquired immunity, and that is what you think about when you vaccinate an animal, which will trigger both cell-mediated and antibody-mediated immunity. And this is a very successful, long-proven method to help control disease. However, with mastitis, there are so many pathogens that it's kind of like the common cold in humans. There are so many things that we would need to vaccinate against, it becomes impractical to vaccinate for all of them. The other branch of the immune system we refer to as the innate immune system. And this aspect of the immune system is a very old, from an evolutionary point of view, aspect of immunity, but it's very, very important because it represents the first cellular line of defense against pathogen invasion. And there are two cell types known as macrophages and neutrophils that are part of this innate immunity that respond very quickly to try to kill pathogens. And we call them phagocytic cells. Phago meaning to eat. Cyte, C-Y-T, is an old term that means cell. So cells that eat. And so they basically try to eat and digest pathogens. We've known this for a very long time. In this slide, I depict a Nobel Prize winner, Dr. Elie Metchnikoff, who was a Russian zoologist. And in his Nobel Prize acceptance speech that he received in 1908, he referred to the battle between a pathogen, he refers to it as a morbid agent, and the external microorganism, and the mobile cells of the organism himself. Now, you got to realize they had very limited capability to study the cells of the immune system at that time. But they recognized that if the cells of the immune system win the battle, that would represent a cure and a victory of the immune system, and that the animal should go on and survive. If not, it may not be such a good outcome for the animal. Curiously, mastitis had a big role in Metchnikoff's early observations. He actually cited in his Nobel Prize speech a Swiss veterinary expert who had been studying mastitis, in particular streptococcal mastitis, And Schottky had determined that if the cells in the milk were capable of eating and destroying the streptococcal organisms, then that was a good thing. However, that phagocytosis, or eating and killing of this bacteria, was insignificant or not adequate, that those cows would be written off as no longer being capable of producing good milk. So we've known for a very long time, this Swiss veterinary expert was doing research on mastitis in the late 1800s. So we've known very long that neutrophils, the phagocytic cells that are the most prominent ones in the milk, are extremely important for protection against mastitis. They're professional phagocytic cells, the first responders, or the predominant cell type in mastitic milk, and they are absolutely essential for udder health. They also are very important for preventing or resolving many of the other diseases in a postpartum cow, such as mastitis, retained placenta, and metritis. And we'll go quickly through some of the data that supports that statement. It's also important to understand, and I put this on here to emphasize the point I made earlier about a high incidence of Johne's disease as well as salmonella in early lactation, that 80% of the neutrophils that are in our bloodstream leave the body through the intestinal tract. And this suggests a role that they play in gut health. Some of my colleagues in Beltsville, Maryland with the USDA many years ago determined that physiologic stress in a lactating dairy cow would reduce neutrophil phagocytosis in milk. And this was work done by a former colleague of mine, Max Papey. And so he had first proposed that stress would enhance the susceptibility of the mammary gland to infection. And he used that by giving some hormones that would trigger the release of cortisol, a well-recognized stress hormone. Another study was done by another colleague, Dr. Jean Lofstedt, who was studying mastitis in sows. Yes, that's the pig, not the cow, but she was doing research where she was able to show that impairment of neutrophil function, the PMN, in sows was associated with naturally occurring E. coli mastitis. And she was able to measure different functional tests of these neutrophils and found that if these things were decreased, that these sows would actually have a much higher rate of E. coli mastitis, which was not a good outcome for the litter. So we began doing research based on those studies to look at what is the immune status of the transition cow. And we first began working with heifers, and then we extended to multiparous cows. And subsequently, we've looked at well over 200 cows over the period of years, doing very intensive studies. These are very challenging studies to do because we separate all the cells out of the blood and then run them through their tests that we do in the lab. And what I show on this slide here is that in the yellow area, that's the week or two around calving. And all three of these tests are measures of how well the neutrophils, those cells in the milk that fight the infections that cause mastitis, they're all experiencing some level of decline around calving. And it makes the cow more susceptible to the clinical disease, especially if she's already infected. This immune impairment is not limited to just the neutrophils. Colleague of mine, Dr. Judy Stable, in one of our studies also evaluated the ability of another cell of the immune system, the lymphocyte, to produce antibodies. And as you can see in this slide, again, the couple of weeks before and after calving, you can see that there's a dramatic impairment in the ability of the lymphocytes to produce antibodies. And we've known this from very practical levels that you may need to vaccinate the cow around calving time, but the typical booster response is not always real evident, but you still need to do it. Another colleagues of ours in Japan, about the same time we were studying what was going on here in the United States in the immune system of a cow, they too were looking at the ability of the lymphocytes, the ones that are involved in antibody production. But those lymphocytes also produce important molecules called cytokines that help activate the immune response. And Ishikawa clearly demonstrated in a study that he published in the early 90s that the production of interferon gamma, a very important cytokine of the immune system, is dramatically impaired. And again, you can see that it declines from levels 5 weeks before calving down already a couple weeks before calving, that it's quite low and then it invariably takes 2 to 3, 4 weeks for it to recover back to a normal level. We extended our work to a very big herd with a collaborator of mine at Iowa State University that we were looking at various factors associated with the genetics of the immune response. And we did find that there is some genetic component to this the immune suppression that the cows experience. But here's 137 cows that we did. And you can see very dramatically that the immune response bottoms out that first week after calving. So you're seeing a pattern there of declines of the immune capabilities of both lymphocytes and neutrophils. And we think that they have very critical importance for the development of disease in those postpartum cows. In fact, there were studies done in England that also showed that if there's any delay in the movement of neutrophils into the mammary gland after calving, and that a process they refer to as diapedesis, meaning that the neutrophils leave the bloodstream, get through the tissues, and end up in the milk to fight the infection. If there's any delay in that process, that those cows developed a very severe form of what we call a peracute case of coliform mastitis in that particular model. And the cows would get very high levels of E. coli in their milk and suffer very severe endotoxemia. And they would often see a very high mortality rate in those cows right after calving. So any sort of delay in that inflammatory response of moving the neutrophils into the milk was a very poor prognosis for the cow. Another study done in the Netherlands by Gunning et al., they also were able to show that if neutrophils from cows were impaired in their ability to migrate, that they would go on to develop retained placenta. In fact, Gunning and his colleagues, they were actually able to predict which cows would develop retained placenta based on impaired migration of the neutrophils in a test that they would run in the laboratory. Now, none of these tests are very practical things for us to use on a farm, but they're very informative in telling us what is wrong with the immune system and what we might consider doing about it. So how do these neutrophils get from the blood into the udder? And I'm going to walk you through a couple of slides here. And what I have here is on the left-hand side, the capture portion. These are a cartoon image of the neutrophils where they're going through the bloodstream. They make contact with the long, flat blue cells, which are the lining cells of the postcapillary venules in your blood cardiovascular system. And as they roll through the capillaries, they're monitoring for any changes that are occurring in that tissue that might be caused by an infection. If they detect any signals that the tissues send out that there's an infection there, those neutrophils will stop, they'll quit rolling, they'll bind tightly, and then they'll gradually migrate between the cells that make up your postcapillary venules and go into the tissue to combat the infection. And I got a video here that we're going to show. This is a video that is actually in a rabbit, but it's a good example of what happens in a cow. And what we've done, and colleagues that have provided me with this video from Boehringer Ingelheim, what they did was they anesthetized a rabbit, and then they've been able to use a microscope to actually look through the mesentery, or part of the gastrointestinal tract that's rather transparent. And they have focused in on an arteriole, which is part of your arterial blood supply, and a venule in the yellow there. And what you're going to see is there's red blood cells that are moving rapidly, so rapidly you don't see them through the arteriole and the venule. But on the venule side, you'll see these cells that the arrows are pointing at. Those are the white blood cells, or mostly neutrophils, that you're going to see that are doing this rolling, monitoring for infection through a molecule known as L-selectin. And then if they sense an area or a tissue that's has activation signals because there's an infection, then they bind tightly with the beta-2 integrins, or the tight adhesion molecules. So in this next video, it's a little bit slowed down and you can't quite see because of the need to limit the size of this file, but you can see that in that top venule, you have the neutrophils moving through. It's a little bit of a herky-jerky movement but they are actually monitoring for any infection in the tissues, stopping momentarily and then releasing. And on the bottom there, the arteriole in the bottom, you see none of this actually happening. So the immune system is very tightly regulated. It only occurs in the venous side of your blood system, cardiovascular system, and that's done to limit any excessive tissue damage. So this video here shows what happens. So this would be very similar to what would be in a dairy cow that has coliform mastitis that may be quite severe. And you see in the bottom venule there, again, on this one, it's upside down compared to the last one, arteriole on the top, but a postcapillary venule on the bottom. You see all these neutrophils have stopped rolling. They are almost plugging the postcapillary venule because they're in the process of migrating out into the tissues to combat the infection. So if you have a severe endotoxemia in a cow with coliform mastitis, this is exactly what that would look like in the venules in the udder. So that's immune surveillance. And it's a very important step in the entire process for the innate immune system to be protecting the mammary gland. And we did a study that we were able to show that those surveillance adhesion molecules, they are shed or lost on the surface of neutrophils right around calving time. And so we have the previous slides that you saw where there's the impairment of the killing mechanisms, the oxidative burst that neutrophils do. And then now we show here that the immune surveillance is dramatically reduced right around the time of calving. So that's two bad strikes against the cow's health because not only are the neutrophils impaired, but they're not doing an efficient job of immune surveillance. One of our other colleagues, Kayoko Kimura, did a project here where she was very focused on the development of retained placenta. And in this graph, on the bottom in the red, you see the ability of the neutrophils to execute the killing activity of myeloperoxidase, a very important enzyme that neutrophils use to kill bacteria. You can see that in 20 cows that would eventually develop retained placenta, that even 2 weeks before calving, they were already showing the beginnings of impairment of the myeloperoxidase enzyme activity compared to cows in the top green bar line that did not. So it's important to understand that this immune impairment begins and it exists well before calving time. Similarly, she looked at migration, the chemotactic activity of the neutrophils, much like Gunnick did in the Netherlands years before. And again, the impairment of the neutrophils' migration capacity existed several days before calving. And those animals would go on to develop retained placenta. And a dramatic difference compared to those cows that did not. Another colleague of ours at the University of Illinois, graduate student Kai, who did his research on transition cows as well, a few years after we began in this. And key thing that he demonstrated was that, again, this is that myeloperoxidase or iodination reaction. The myeloperoxidase activity was impaired the most in cows that would go on to develop retained placenta membranes, cows that would develop metritis in the red line, and then the cows that developed mastitis was in the black dashed line. So the cows that got more disease problems were the ones where their neutrophils were most impaired. So correlating the degree of immune impairment with the actual onset of disease. So that immune suppression summary is that the effects are transient. They last several days to a few weeks. They delay the inflammatory response by reducing the efficiency of immune surveillance and egress or movement of the neutrophils out of the blood into the tissue to combat infection. And even if they do get to the infection site, their function is impaired. Oops, I get back here. The lymphocyte capacity, not to ignore the importance of the adaptive immune system, is also impaired or delayed and will affect the animal's response to vaccines. So both the innate and adaptive immune systems or acquired immunity are impaired. So Mike, I'll turn it over to you here.
Yeah, we have our first polling question, so get ready to vote here. And we're off and running. Jim has opened the poll. Select each statement that's true, and I assume more than one correct answer are possible. Number one, mastitis mainly is an opportunistic infection. Number two, innate immunity is a key protection against mastitis. Number three, transition cow immunity suppressed starts prior to calving. Number four, the greatest risk of getting infected is right after calving. And number five, 50% clinical mastitis is in the first month of lactation. So we're off and running here, and boy, we're a little slow on voting. This is kind of a challenging question, Dr. Curley, that you had here. Abby, are you going to vote?
If I was picking them, I think I would select all of them. Some of the wording maybe is tricky. Dr. Curley, you'll have to go through in a little more detail, but just as I look through a couple of them, I feel like they're accurate. So I would lean towards saying that they all are true. What do you think, Mike?
Yeah, I would vote that way too, all and true. And we're up to a minute, and we've got over 60% of the vote. So, Jim, let's close the vote. And it's kind of interesting to see the numbers. Dr. Curley, what do you think? What's your take on the vote?
Well, it appears that I needed to make a better emphasis on the fact that the cows get the greatest risk of being infected right before calving. But I think the general message, yes, they were all true. It is an opportunistic infection. Innate immunity is vital to the protection against mastitis. The impairment begins prior to calving, and you get nearly half of the clinical case of mastitis in the first month of lactation. So all true. All right. So what can we do about bolstering the immunity? Can you modulate those neutrophils to enhance the cow's resistance to mastitis? Some of the first studies that were ever done on this were colleagues again in Beltsville, Maryland, and they used these plastic intramammary polyethylene devices that they put up into the teat, and these serve to raise the somatic cell count in the milk, or the neutrophils. While it was very effective, they had some downsides because they would cause a little bit of bleeding, and you get some pink milk from time to time, and some of them would come out during the milking process. So while they turned out not to be a practical solution for mastitis prevention or reduction, they did establish a good proof of concept that if you increase the number of neutrophils in the milk, that you may actually reduce the incidence of mastitis. Of course, that's a real challenge because you want to keep your somatic cell counts low. So it had some impractical aspects to it as well. But they were mainly looking at the cell counts that were in the foremilk, or not the main amount of milk that the cow would produce while being milked. But we began to ask the question, can we use biotherapeutics or a different approach to increase the circulating pool of neutrophils that are important for mastitis resistance, and even maybe enhance their function right around calving time? So we wanted to restore that dysfunctional immune capability that happens around calving time. Didn't want to take it too far. We view that this is a prophylactic approach, and that it's important to understand immune modulators require some time to restore that impaired or underdeveloped immune capacity. And immune modulators are very different than an antibiotic. They have immune-specific mechanisms of action rather than a direct antibacterial effect. So one of the molecules that we all settled on, myself and a couple of other colleagues in California and one down at the Hill Farm in Louisiana, was to examine a molecule known as granulocyte colony stimulatory factor. This is a natural molecule of the immune system of all animals, and its primary role is to increase production of neutrophils from the bone marrow where they're normally made. And there's a lot of now published literature on this molecule, and it activates a critical adhesion molecule that would predict faster response, but more efficient immune surveillance. And it also enhances a range of neutrophil functions. So we began evaluating that molecule and found that depending on the dose you would give, you'd get an increase in the number of circulating neutrophils that was very dose dependent and predictable. And this is our first study we did here back in the late 80s now, where we dosed some prepartum dairy cows, and we began treating them roughly 2 weeks before calving. And you can see, and this was a very early study where we were giving daily injections, And it took about 10 days for the cows to reach their peak response at this particular dose that we gave. But it was a very profound effect on the immune system. And you can see that the normal count was down around maybe 2,500 per microliter of blood. And we got them up to around 45,000 on average. So one of our colleagues, Dr. Jim Culler at UC Davis, he actually was involved with some of this very early work, as was Steve Nickerson down at the Hill Farm in Louisiana. In Steve's work, he pre-treated cows with GCSF and then would challenge them with Staph aureus. And he was able to show a reduction in experimental Staph aureus mastitis by 46, almost 47% reduction. And then Jim Culler used Klebsiella, one of the severe coliform mastitis pathogens. And in his study, again, he pre-treated cows with GCSF and then challenged them. And in his study, he looked at the infection status. The control cows that were challenged, they were culture positive with Klebsiella for a week, 7 days. And the cows that he treated with GCSF, they cleared the infection by 6 hours. A very dramatic observation. And clinically, those cows in Jim's study, the controls developed a fever, severe fever, and abnormal milk for 5 days. The cows that received GCSF were no longer running a fever after 12 hours following challenge, and their milk was normal in appearance within 24 hours. So some very profound impacts on cows, and both of those studies were done in mid-lactation cows. So we went on to evaluate GCSF in a study that we did in early lactation cows, where we started giving it 3 days after calving. And in the study that we did here, we did a very low dose. It was only 1 microgram per kilogram. And started giving it daily. We treated them for 5 days in a row. 3 days into the treatment regime, we challenged them with E. coli in 1/4 and monitored the outcome. And in this study, we used human GCSF because that was what was available at the time. Well, we also saw a 50% reduction in the number of new infections that took from our challenge. Normally we get 100% take. We saw faster bacterial clearance rates, reduced severity scores. Cows had improved milk production and feed consumption during the course of our study. And the cows that got the saline, it took them almost 4 days to clear the infection, where the cows that received GCSF cleared it within 30 hours. So lower dose than what Jim had used, but Mike, I'll turn it over to you, and we'll see what the response is to these questions.
All right, here we're off and running again. Dr. Curley, effectively restoration of the immune function can be, and select all that are true. Number 1, reduce the incidence of infectious diseases. The second choice, reduce severity and duration of infectious diseases. Number 3, reduce antibiotic use to treat mastitis. Number 4, reduce the amount of milk discarded, increase milk sold. So which one of these are true? You can go for more than one. And Abby, how are you going to vote here for the effective restoration of immune function? Which one are you going to select?
Yes, Mike, I think an interesting solution, potential solution to mastitis. I think I, once again, would pick all 4 choices as some of the benefits of effective restoration of immune function. What do you think?
Yeah, I'm going to vote with you again. I think I would take them all as far as that goes. Be interested if some is more, Dr. Curley, more important than others. But we're going to close the polls because we've got 2/3 of the vote in. And what do you think of the results, Dr. Curley?
Well, it looks like everybody was paying attention. So yes, each answer would be true. You get a reduced incidence of infectious disease such as mastitis. We reduce both the severity and duration of those that do occur. If you still get the infection, we do reduce antibiotics because you got less mastitis to treat. And the amount of milk discard is reduced, and you, in essence, then sell more milk. All right. So the next part of this is going to be talking a little bit about what we can now do. And in 2016, whoops, went too far here. The FDA approved a new product, which is a form of this GCSF. It's called Peg-Boviglastim. And what I'm presenting here is some published work that they reported. This Peg-Boviglastim is for use in transition cows First dose to be given about 7 days before calving, and then a second dose to be given within a week of or within 24 hours of calving. And in 4 U.S. dairies where they conducted this work, they had 320 cows per treatment across those 4 dairies. They got an increase in the circulating number of neutrophils within 24 hours of the treatment. They got a 35% reduction in the incidence of clinical mastitis, even though they only treated 7 days before calving, and within 24 hours of calving, there was a lingering effect to reduce clinical mastitis during the first 30 days of milk. And it also improved the return to estrus, reduced the failure to return to estrus as measured over the first 80 days in milk. So that was a very interesting finding. They evaluated their product after it was launched in Mexico on over 5,000 cows in 17 Mexican dairies. In these dairies, they had a fairly low incidence naturally of mastitis during the course of their study, but they still reduced the incidence of mastitis by 25%, reduced the number of treatments. They had less discarded milk, reduced retained placenta, and improved the odds of getting their cows inseminated during the first 100 days in milk. So some more recent work that people have been looking at is that in this particular study here, some colleagues of ours looked at what would happen with this same product given to mid-lactation cows. And in this slide here, in the blue line, you'll see the increase in the neutrophil counts. With the first dose there, you get a nice little peak there within about 4 days after giving the compound. And then they repeated it a week later after the first dose, and you see a very dramatic increase where that neutrophil count went up to over 50,000 neutrophils per microliter of blood on average. So there was like a twofold increase. And I think what's going on here is the first shot kind of gets the bone marrow turned on, you get your increase, and then that second shot really expands what's going on in the bone marrow and really gets that cow in a position where she's producing a lot of neutrophils to help keep her healthy. And you can see the effect lasts out for a good week to 10 days after, or almost 2 weeks after that second infection, where you still have an elevated neutrophil count to help protect the cow against infections. In that same study, they went on to do a challenge of cows with E. coli in mid-lactation. And similar to the work that Jim Culler and Steve Nickerson did with mid-lactation cows, the cows that received GCSF in the blue line, they had fewer bacteria in the milk after they were challenged if they had been treated with the peg bovine stem. So it reduced the severity of the disease and the number of bacteria in the udder that the cow had to combat. They also took a new approach at looking at this myeloperoxidase enzyme, which is the most potent oxidative killing mechanism that neutrophils have. And on this graph over here on the far right of the slide, in the blue, you see there's a shift to the right, which means there's an increase in the amount of this myeloperoxidase enzyme that is expressed on the neutrophils. And this is perceived to be a very positive outcome. First time this has been reported, I believe, in any species. And in addition to being a potent killing mechanism of the neutrophil, this myeloperoxidase is now also perceived to play a very important role in migration of neutrophils into tissue. So presumably all positive for helping the cow. Also, earlier within the last 6 months, there's been another paper that came out of Germany evaluating the effect of peg bovine stem on postpartum cow health, where they focused on metritis in cows. And this was a fairly large study. They started with over 80 cows per treatment group. And what they found was that they were able to reduce the incidence of acute puerperal mastitis by 48% in the cows that received the label dose of peg bovine stem. And in addition, they reduced the number of antibiotic doses. They cut the number of antibiotic doses per calving nearly in half in the cows that received GCSF. So with GCSF, there's no milk discard requirement. So anytime you can reduce antibiotic usage, you're reducing the amount of milk you have to discard. So it's all good. So today we've reviewed that this immune suppression is a problem for the cow. It has many causes, and I didn't try to get into those today. but many physiologic transitions such as calving, shipping, transport, weaning are all factors that will stress a cow's immune system or a calf's immune system. And today, since 2016 in the United States, there's been an FDA-approved immune restorative available that very much targets the most important immune cell, the neutrophil, for the protection of the udder. So I think it's very important to focus on your transition cow management on your farm. And I'm sure we still need more research to get this problem licked. But with that, Mike, I'll turn it over to you again.
Last polling question. So which of the following are true? Giving birth is extremely immunosuppressive. Second choice, reducing transition cow exposure to pathogens, a good idea. Number 3, clean, dry conditions, the dry lot are not important. Number 4, mastitis before peak lactation will blunt the lactation curve. Number 5, there's nothing we can do to help the transition cow. Okay, Abby, be careful on this one. I think we could be excited. What on this one?
Few trick answers in this one. I am going to say the following are true. Giving birth is extremely immune suppressive. Reducing transition cow exposure to pathogens is a good idea. And mastitis before peak lactation will blunt the lactation curve. I'm picking those 3. What do you think, Mike?
I think I guess I vote with you. We'll see if we're wrong or not here in just a few minutes. And those few minutes are done now. 2/3 are in. Jim, let's share the results. And Dr. Curley, what do you think on the results?
I think everybody was paying real close attention, that or they're paying even more closer attention on their farm, because I think any dairy farmer that follows their cows closely, they understand that it's a very immune suppressive event to give birth. And that keeping the exposure to pathogens in those transition cows is extremely important. So I was happy to see that. And it's also well recognized that if you have a case of mastitis before the peak of lactation, you will blunt the entire lactation curve. So preventing mastitis has a profound financial impact on a dairy farmer's bottom line. So with that, I'd like to close on this thought here that we can improve on California teat dipping. And this is definitely not what we want our close-up pen or our dry cow lots to look like, much less any of our lactating cows. And I got this picture from a photographer who was advertising in Hoard's Dairyman more than almost 30 years ago. And I don't remember his name at the moment, but it was a very profound observation he made driving down a highway in California, and he noticed this picture. So I appreciated his power of observation that this type of teat dipping needs to be reduced.
Okay, Abby, I think you can maybe do a preliminary summary before or comments before we move into the Q&A period.
I sure will. Thanks, Mike. Thank you, Dr. Curley, for that very thought-provoking, informative presentation. And I think that final slide is also very memorable for most people, something we just don't see very often, and hopefully we'll never see, but it's just a reminder of how all the details can really make a huge difference in cow health and transition cow care. So thank you for leaving us with some new kind of tips and tricks to hopefully booster that transition cow immunity. Definitely want to thank Dairyman V for sponsoring this session. If you want to view this webinar again or any of our previous webinars, you can go ahead and visit our archives. Our website is at www.hoards.com and then go to the webinar tab. Also, you will be receiving an email survey later this week. Please fill that out, as that helps give us ideas for future webinars and helps us improve anything that we can to make these an even better experience for you. Please, we hope that you will make plans to attend our next webinar, which will take place on Monday, October 8th at noon Central Time. I think if we click back to that slide, you guys can see the presentation titles there. In October, we'll be talking about the feed supply and the quality cards we've been dealt. Before the webinar, we touched briefly on kind of the wet conditions in some areas and what that will mean for the harvest season. Well, if we get together in a month, we'll be able to kind of talk about what we're seeing so far and what's up ahead for the feeding in 2018 and 2019. So our presenters for that will be Mike Hutchins from the University of Illinois, and then Mike Rankin, who is our Hay and Forage Grower Magazine managing editor. That presentation will be sponsored by Kuhn. Looking ahead even further, our November webinar will take place on November 12th, and same time, same place. And that presenter will be Larry Trainell from Iowa State University, and he will be talking about user experiences with robotic milking, some of his experience working with producers who have switched to robots, plus some surveys that they've done with Iowa State University Extension. So please make plans to attend that webinar as well. I think a few questions have come in while you were presenting, Dr. Curley. So Mike, if you want to go through any questions, and let's let Dr. Curley take a stab at answering those.
Okay, well, let's go to the one that came in early, Dr. Curley. You want to, the question is, how to evaluate practical level immunity on the transition cows? We have problems of mortality. This is an international question, I believe. Dry cows after they're drying off, definitely connected to immunity level. What could you possibly pose for farmers in this kind of situation?
Well, this is always the most difficult question that anybody ever asks me is how can I assess the immune status of the cows on my farm? And it's not easy to do, and I wish I was smart enough to develop a simple test that you could run on your cows to know which ones are most immune suppressed. But the bottom line is the laboratory work that people around the world that have looked at this immune capability of a transition cow, very expensive projects. And they just really are not something that anybody's been able to reduce to a practical level, because you're looking at many aspects of the immune system. The cells, the different types of cells, different capabilities that we can measure in the lab to look at the whole picture of the immune status. And it can't really be reduced to one simple test like the level of antibodies. That's really not going to do you much good when it comes to predicting resistance to mastitis, for example, or metritis or retained placentas. So it's very, very difficult. And I don't have a good way of having anybody assess that, short of getting involved in a massive research project, which nobody on a dairy farm is going to want to do. The latter part of that, problems of mortality in dry cows after dry off, I don't want to commit malpractice here. This is one of those situations where I think a veterinarian would want to be right on the farm and understand, okay, what were the diagnoses? What were the actual mortality causes? Because a lot of things can cause a mortality. And I think it's very important to understand what the pathogens were, what were the actual cause of death. So I have no doubt that your assumption that it's connected with immunity is probably correct. But the best thing to do would be to engage with your veterinarian on your farm and do as much laboratory workup of what's killing your cows and have them investigate where the evidence leads them.
Dr. Curley, let's build on his question a little bit further. What are 3 or 4 things you would recommend if you said, I want to really improve the immunity in my herd other than vaccination program? Are there anything, well, maybe vaccination is it, but is there anything nutritionally speaking or environmentally that you would come right to the top of your list of saying, do these things?
Well, as we alluded to in the talk, keeping your transition cows in the close-up pen, your dry cow lot, keeping them dry and minimizing pathogen exposure is extremely important. And that I know has often been an area that we don't tend to pay as much attention to our dry cows as we should. I am not a nutritionist, nor do I play one on TV. However, work closely with your nutritionist to make sure that you're avoiding metabolic diseases that occur during the transition period. It's well recognized that things like milk fever have an important or a very important contribution to the development of postpartum infectious diseases. And if you can avoid milk fever, you're going to make a significant gain on keeping the cows healthy. They still become immune suppressed, but maybe not as severe and perhaps not as long after calving. The other thing, there's been a lot of research on nutrition, trying to identify ways to enhance the immune status. Perhaps the most profound evidence that supports proper nutrition is the body of work that's been done on vitamin E and selenium and its relationship to the cow's resistance to mastitis. We know that if you maintain adequate levels of vitamin E and selenium in the diet, that you will reduce the incidence of mastitis, but hypersupplementation of either of those is of minimal to no value. So making sure that the key nutrients in the ration are at the proper levels is extremely important, and understanding where your herd is at, work with your nutritionist, is extremely important. Other nutritional factors, it's been very difficult to show that beyond vitamin E, selenium, they're perhaps the best examples where you can get measurable effects on the immune system by having adequate vitamin E and selenium. However, other nutritional approaches, there's very little published data with statistically significant effects on the immune system that result in reductions of clinical disease. So that's a very high bar to get over if you're trying to modulate the immune system nutritionally. And that's why most of the research is focused on factors associated directly with the immune system, like GCSF, because we know we're absolutely targeting the most important part of the immune system for that transition cow. So I don't know if I got all of your question answered there, Mike, but that's kind of my view on it.
Yeah, I think you've picked up a couple of the other questions we had here on trace minerals and stuff like that as well. Here's one for you, though. It says, if FDA has approved, and I'm going to use the word PEG, which is the long, short name of that, why is not been taken off the United States if it's approved here? Obviously, they're using it in Mexico. Comments on why we don't see more of it being used in the U.S. based on your data?
Yeah, I've just recently been made aware that they're going back to research on that. I'm not directly involved with that product or what they're doing there. I'm not sure if they're trying to get additional label claims or what the purpose of that is. So I really can't answer that question. I think the scientific data does support its use, but there's always other factors that maybe are involved that are related to the practical use of it. I really can't answer that question. I wish I did.
Yeah, is it commercially on the market? Is it available if I wanted to buy it, or is it even available in the U.S.? Do you know?
My understanding is it's available in the U.S. through the end of this calendar year.
Okay. What about mycotoxins? Do you think mycotoxins could have an impact on the immunity around calving?
Absolutely. I think mycotoxins are an extremely important thing, and that probably is going to very much relate to your topic next month, Mike, on the quality of our feedstuffs. I think that we have to be very, very careful with what's happening with our feedstuffs. And I think there's also considerable concern around what we might call just subtoxic levels of the mycotoxins, because there's so many of them. And what if you have just borderline levels of 3 out of 4 or 5 mycotoxins? Might that be very different than just elevated levels of 1? And so managing the quality of your feedstuffs to minimize molds getting into your feed and the mycotoxins that are produced therein, that is extremely important. And we've known for years that mycotoxins will throw cows off feed. They can raise liver enzymes, even if they're not showing a lot of clinical symptoms. We knew that with the fumonisins many years ago. And I think that that issue is an area that needs a lot more research than the attention that's been given.
Okay. Yeah, I've got to move up my slide here so we make sure we have visibility as far as that goes. Another question that comes across, and that is low somatic cell counts in cows, and yet it looks like that's where some of the immunity is going to come from. What's your take is having really low somatic cell counts, down, say, 50 or 60,000, really low? Are these cows better or worse off from immunity?
Well, I think that when you have your cell counts that low, that's a good indication that your environmental exposure to pathogens for those cows is very low. What's the most critical thing is, are those cows capable of responding and increasing that somatic cell count in the first few hours after they're infected? I didn't show, but organisms like E. coli and most of the organisms that cause mastitis, they love to grow in milk. You can grow them in milk almost as fast as you can grow them in any media in the laboratory. And E. coli in milk will double every 2 hours. And so it's very important to have that very rapid immune response. So whatever your baseline is, if you're at 50,000, you're doing a fantastic job of milking your cows, but they can still get mastitis. And so the key is, how quickly can that cow respond? If she's got a large pool of neutrophils in the bloodstream, that can respond more quickly, and you can get more cells in there in that first couple of hours after the infection, you're going to reduce that bacterial burden that she has to overall deal with. So to me, that's the more important measure, is taking that 50,000 and getting it up to, let's say, 500,000 very transiently and quickly, so that it kills off all the bacteria and goes back down to your lower level within a few days. So that's the most important thing. And it's not something that you're necessarily going to be able to detect with your somatic cell count that you get from DHI.
We have a couple of international questions come in here that are kind of long, but I'll shorten them down for you a little bit, Dr. Curley. 3,300 cow dairy herd here. They're doing all the things with anionic salts, vitamins, selenium, protein, energies, looks good. Plenty of bunk space here. What else can they do to reduce retained placentas below 10%? So any thoughts on that? It's kind of a kitchen sink type question.
Yeah, that's a little bit, again, I'd need to know a little bit more about what's going on that farm that might be contributing to high incidence of retained placenta. They have a lot of twins or what else may be going on in that herd. Sounds like they're doing a lot of things right. If they have access to pig bovigrastim, then, that's something that they may want to take a look at. Right now, we don't have a whole lot of other immune modulators available to us. So I really can't offer anything at this point in time.
I think you've answered this question already, but if we reinforce or improve the immune system, will we have a problem with higher white blood cell count in the milk? Thoughts on that?
Sure. That's probably been always the number one question that I've always been asked when talking about use of GCSF in dairy cows. The evidence that was published on pig and bovigrastim shows that, according to the label use, it does not increase the somatic cell count in the milk. You're raising it in the bloodstream in the cows, and then they have a quicker response to a mastitis pathogen, or if you're concerned about what's going on in the uterus, a quicker response to combat metritis and retained placenta. I think it's important also to understand, if you were to give a higher dose of pig and bovigrastim than label, you may eventually get to a point where you would raise the somatic cell count. However, that would pale in comparison to the millions that you get per ml of milk in a cow with mastitis. So clinical mastitis with coliforms, you're going to get somatic cell counts well in excess of 1 million. And, using a cytokine like GCSF in some of our research at doses that were higher than what pig and bovigrastim is used at, we did raise the somatic cell count, but it was for a transient period of time. You got to remember, in your cows after calving, those first 8 milkings you're supposed to be discarding for the pasteurized milk ordinance. And so even if they are elevated in that first day or two after calving, you're feeding that colostrum to calves and not putting it in your bulk tank. So it really is not likely to have any impact on your bulk tank.
Okay, here's your polling quiz coming in from Slovenia, and that is, we're going to list 4 things, and which one do you think is most important to booster transition immunity? Number 1, transition grouping strategies, close up, far off cows. Number 2, a dry, clean environment. Number 3, feed additives or antioxidants in the transition ration. Number 4, a low energy diet with high dry matter intakes in the entire dry period, kind of the high straw diet. So if you only could pick one of those, which one would you pick, Dr. Curley? I think that's the question.
Well, they're all good things to do. I think that in the absence of being able to perfectly manage your nutrition, which is always a challenge, I would reduce that pathogen load. So keeping that dry cow lot as clean and well managed for those cows as you possibly can, because even then, if their immune system is off, if they got fewer pathogens to battle, the odds are in the cow's favor.
Okay, what about injectable vitamin E and A, D, and E during the dry period or in transition period? Any thoughts on injecting those vitamins and products versus probably feeding them? Or probably, I think you already answered, you're a proponent of the vitamin E and selenium. I think they're asking about route of administration.
I would defer to your nutritionist as to what they feel works best on your farm. I think it's always best if you don't have to be managing the cows and treating them with injectables. So if you can get that done through the ration, I think that's probably your best way to manage that. But falling short of that, if you need to use an injectable, certainly do it.
Okay. Have other a couple comments here. People said they follow certain companies' feeding lines, and we understand where that goes here. So we already mentioned the cost effectiveness of the use of the product that you mentioned, the PEG product, as far as that goes. Do you have a cost on that? I think they're asking about that product. Do you know what it costs to those 2 injections that you've done some research with?
So I don't have the exact current price, but I think a ballpark figure today for the 2 doses combined cost was somewhere around $15 US.
Okay, very good. And there's also a question here coming with a commercial product that claims some immune stimulation, but if you stop using it, is there a risk there associated with stopping that practice or not? In other words, some type of immune stimulator being fed or injected animal, and then you stop it. Is the cow going to rebound, or cow's going to rebound, or good news, bad news?
Yeah, so I don't know if they're referring to some specific immune modulator. I do know that when the cows go off of GCSF, they just return back to their normal levels. There's been no detectable negative impact of the extra treatment with GCSF going away. So whether there's other immune modulators out there that are being used in various locations, there's only one other immune stimulant that I'm aware of, and its label claim is for bovine respiratory disease, and I'm not aware of any negative impact of no longer using it after people are putting it in there for reduction of and prevention of bovine respiratory disease. So I suppose there's always something out there that a withdrawal would be a problem, but I am not aware of anything at this time.
Here's your last question that just came in. What effect would vaccination at dry off or during the dry period have on the general immunity during the transition period?
Well, it depends what you're vaccinating against. If you're vaccinating it with mastitis in mind, the main vaccine that has good data supporting its use is the endotoxin vaccines like J5. And the J5 vaccine approach protects against the lethal effects of endotoxemia. It does not necessarily reduce the incidence of coliform mastitis, but it protects against the damaging effects of endotoxin that the cow has to combat. So to me, the evidence suggests that you absolutely need to give a cow the number of shots on the label. Certainly follow that, and I know some of them, they require a booster at calving time. And so whatever your vet's recommending for your herd health program, please stick with it, because they know the problems on your specific herd better than I would ever be able to do here remotely. So yes, you need to maintain your vaccination programs, because you're actually looking at a wide range of diseases rather than just mastitis alone.
So now you're going to answer my question, and that is inflammation. Is that good or bad? If a cow has inflammation, is that good or bad as far as the health, the immunity response in the cow?
Yeah, so it's one of these double-edged swords. You want inflammation to kick in to combat the infection early and efficiently when an animal becomes infected. So inflammation is very important to have it happen quickly and efficiently. You don't want it to drag on long, and you usually see that when there's a delay in inflammation starting. So it, for the most part, properly executed inflammatory response by the immune system is exactly what you want. Ideally, you don't want an infection, but it's hard to keep cows in a sterile world. So having a very efficient inflammatory response is your number one priority. And by efficient, it means it kicks in, it combats the infection, eradicates it, and returns back to normal status, Mike. So that's the best way to think of that.
Very good. Well, listen, we've answered all the questions at this time, Dr. Curley. I appreciate your professional responses and excellent presentation. Abby, back to you to wrap up our webinar.
Thank you, Mike. Definitely. Thank you, Dr. Curley, for taking the time to answer those questions. And we really appreciate all of you who are listening out there, taking in the webinar, and then also submitting some questions to be answered by our speakers. So thank you for taking the time to join us today. We want to once again recognize Diamond V for partnering with us, sponsoring this webinar. We definitely appreciate their support. And if you want to learn anything more about their company, feel free to check them out online, or if you have a local representative near you that you can reach out to, please do that. We will look forward to getting together again a month from now, October 8th at noon Central Time. We will be talking about the 2018 harvest season with Mike Hutchins and Mike Rankin. So we hope that you will join us for that. And then again in November with Larry Trainell from Iowa State University, who will be doing that webinar for us. Until then, we'd like to say goodbye to all of our listeners from all of us here at Hordes Dairyman and the University of Illinois. We really appreciate you taking the time to be with us today.